السلام عليكم. أنا الطالب ركان حداد من كلية الطب في الجامعة الأردنية. Today we're going to be discussing the 6th lecture of neoplasia. Hopefully this is going to be a really short, quick, easy lecture. We're going to proceed on talking on specific mutations that cause cancers. First of all, we're going to focus on the transforming growth factor beta pathway. We must understand transforming growth factor is part of a family of cytokines. And this transforming growth factor actually will inhibit the cellular proliferation for specific epithelial cells, endothelial cells, and hematopoietic cells. It is really important in the regulation of proliferation. Okay, it is going to basically stop it when not needed. تمام? Anyways, transforming growth factor beta is a member of a family of dimeric growth factors. Dimeric means it consists of 2 units. Okay. The transforming growth factor beta family includes the bone morphogenetic proteins and activins. They're both cytokines, okay? In most normal, we're focusing on normal cells, epithelial cells, we know them, endothelial cells, blood vessels, hematopoietic cells. شو يعني hematopoietic? Hematopoietic هم عبارة عن stem cells for blood. They're just basically stem cells for blood. So basically, they are progenitor cells that will form different blood cells, white blood cells, red blood cells, whatever. هذول هم hematopoietic cells. Transforming growth factor beta acts as a potent inhibitor of proliferation. This is normally, okay? So normally, it acts as an anti-mitogenic factor, okay? It regulates the cellular processes by binding to a complex composed of the transforming growth factor receptors. مين هم؟ Transforming growth factor receptor 1 and 2. تمام? What is going to happen? Dimerization of the receptor. When will the dimerization occur? When the ligand binds. Okay? So basically it's just as we learned in cytology. يعني. The ligand binds. And this will cause the receptors to dimerize, okay? So this part will move until it dimerizes, with this part creating the actual receptor, okay? So when the ligand binds, the receptor is going to dimerize, okay? And this dimerization and the ligand binding is going to cause a cascade of events, يعني a pathway is going to occur, an internal pathway in the cell, signals are going to be transduced, and they're going to work on inhibiting cellular proliferation. كيف? It's going to activate CDKIs, okay? It's going to activate inhibitors, okay? أي inhibitors? Cyclin-dependent kinase inhibitors. برضو with growth suppressing activity as well as repression of growth promoting genes like MYC and CDK4. إذن transforming growth factor شو اللي بيعمله? Basically, it binds to a ligand, okay? It binds to a receptor, causing the dimerization of the receptor. This is one part, and this is the other part. This is the dimerization. It is going to send signals as a cascade form. It's going to reach the nucleus. Okay, it's going to reach the nucleus. And inside, it's going to work on methylene. It's going to increase the amount of CDKIs, cyclin-dependent kinase inhibitors. It is also going to repress growth-promoting genes. Okay, يعني it activates or stimulates inhibitors and deactivates or inactivates خلينا نحكي ايش اللي هم خلينا نحكي things that cause growth like the MYC gene and like the CDK4. In many forms of cancer, the growth inhibiting effects of the transforming growth factor beta pathways are impaired by mutations that affect the signaling. Okay, so in cancer, these growth inhibiting effects of the transforming growth factor are going to be impaired. حيخربوا. ليش حيخربوا؟ لأنه صارت mutation. These mutations affected the signaling. Okay? So basically the signaling wasn't complete. If the signaling wasn't complete or if something went wrong, this means that there will be an excessive amount of uncontrolled growth كمان. Okay? The mutations that occur will either affect the receptor, and we will focus on the type 2 receptor. Okay, it will alter the type 2 receptor, or the SMAD, the SMAD molecules, they are the transducers. Okay, when the signal is sent from the receptor to the nucleus, it's sent by molecules. These molecules are called SMAD, like SMAD 1, 2, whatever. Okay, they act as, let's say, transcription factors and whatever that will affect the gene expression. So if a mutation occurs to them, the signal will not be transduced and growth is going to be overexpressed. أو خلينا نحكي growth is going to be dominant or uncontrolled or unregulated, whatever you want to say. Okay? ليش؟ لأنه ال TGF beta or the SMAD molecules, they focus on anti-proliferative signals. Okay? Any mutation will basically remove that anti-part. حيصير عبارة عندك proliferative signals. Okay? فيعني it's really a simple thing to understand. Mutation that occurs ruins the protective effect and ruins the خلينا نحكي the inhibitory أو خلينا نحكي the inhibiting effect of growth, which will cause unregulated growth of the cells or unregulated proliferation of the cells. Okay? The mutations in the type 2 receptor, and we'll talk about the SMAD receptor as well, are seen in colon cancer, stomach cancer, endometrial cancer, and pancreatic cancer. Okay? With pancreatic cancer, we'll also see mutational inactivation of the SMAD4. Loss of the transforming growth factor beta mediated growth control occurs at a level downstream of the core اللي هو transforming growth factor signaling pathway. كيف يعني؟ يعني basically when we say downstream of the transforming growth factor, the transforming growth factor binds to the receptor up top. Okay? Downstream means the transducers on the inside. Okay, these transducers, like the SMAD molecules, like the genes that are going to be affected, like other tumor suppressor genes, like other inhibiting molecules like P21, all of these can get inhibited. تمام? So when you lose the mediated growth control, you're basically at risk of losing P21 expression or overexpression of specific growth genes like MYC. Okay, so بكل بساطة, a mutation to the transforming growth factor beta, what is it going to cause? It's going to cause the loss of expression of P21. P21 is a CDKI. It's a cyclin-dependent kinase inhibitor. And or overexpression of MYC. MYC is for growth, okay? And proliferation. It's a proliferative gene. When it's overexpressed, there will be unregulated proliferation. تمام? These tumor cells can then use other elements of the transforming growth factor beta induced program, including immune system suppression or promotion of angiogenesis to facilitate tumor progression. Okay, look at the tumor now. It's going to start using the natural elements of a transforming growth factor. We took transforming growth factor طبعا in the inflammation lectures and pathology as well. We must understand that here, the cancer or the tumor is going to do something that is really bad. It's going to start using the transforming growth factor. Why? So it can evade the immune system. Okay? It will cause the immune system suppression, as well as it will start promoting angiogenesis to itself. The tumor nafso, we already know that cancers are going to start, and tumors are either going to start generating or they're going to start undergoing angiogenesis for more nutrients for it. Okay, they're going to start using the body for its nutrients. Okay, especially by angiogenesis. فبيحكي لك إنه the tumor cells will use the other elements of the transforming growth factor. Okay, they will take advantage of it, including the immune system suppression and promotion of angiogenesis, and it will facilitate the tumor progression. تمام؟ هلا, the transforming growth factor beta has basically two functions. واحد a normal function, واحد cancerous function. Normal function is in normal cells. What will it do? What is it going to do? It's going to prevent the tumor growth. لكن if it got mutated or if a mutation occurs, basically it's going to promote the tumor growth. Okay? So depending on the genes in the cell, are they mutated, are they normal, and so on. Okay. Now let's talk about contact inhibition, NF2 and APC. شو هذا التخبيص انت قاعد تحكي? We'll talk about them in a bit now. When cancer cells are grown in the lab, their proliferation fails to be inhibited when they come in contact with each other, because they do not care about the density of cells. They do not care about how many cells they're being in contact with. That's why a tumor can grow to a really, really large size. Okay? So this means that they do not care about contact inhibition. They don't have anything such as contact inhibition. Okay, they keep on growing and growing and growing without paying attention to what's happening around them. Okay, so here they're just telling me, when we grow them in the lab, they're just going to keep on growing, not caring about anything. And this comes in contrast to non-transformed cells, normal cells. They will stop proliferating once they form confluent monolayers, مثلا skin. When a skin is wounded, the wound is going to close, and after that, no cells or no proliferation occurs to the skin. Okay, it enters a quiescent state. The cells won't keep on growing and forming a mass outside of your skin. تمام? Okay. Cell-to-cell contact in many tissues are mediated by homodimeric interactions between transmembrane proteins called caderins. Okay. شو التخبيص اللي حكيناه؟ Here's a cell, an epithelial cell. Here's another cell, برضو epithelial cell. Okay. Here is a specific transmembrane protein, هذا اسمه E-cadherin. Okay. This is E-cadherin. And this is another one called E-cadherin. Okay. I'm going to write E-cad. Okay. E-cad. تمام. What's going to happen here is a homodimeric interaction. شو يعني homodimeric? Homo means from the same type. يعني E-cadherin من هون, E-cadherin من هون. E-cadherin from each side. Homodimeric means that these together as a dimer are going to work. Okay, homodimeric interactions. هذول هم الcaderins. تمام? E-caderins mediates the cell-to-cell contact in the epithelial layers by two mechanisms. Okay, one of them basically is mediated by the tumor suppressor gene, the neurofibromin 2. Let's focus on the other by using a beta-catenin. Beta-catenin is a protein bound to the E-cadherin from the inside of the cell. تمام? Beta-catenin هون, let's name it beta or B-cat. Okay, B-cat. Beta-catenin has the ability to act as a transcription factor. It travels to the nucleus. Okay, نفس الشيء, travels to the nucleus. And it will cause proliferation when needed. يعني when a specific signal is received, okay, proliferation is going to occur. Okay, المهم شرحنا هون شو يعني homodimeric interaction. We're going to go back to this in a picture that I have on the last on the last page. One mechanism حكينا is mediated by the tumor suppressor gene neurofibromin 2 called Merlin. It acts downstream of E-cadherin in a signaling pathway that helps for maintenance of contact inhibition. تمام? A homozygous loss of NF2 is known to cause certain neural tumors as neurofibromatosis type 2. Homozygous loss, you're losing both of the genes that are normal, okay? So the loss of NF2, what does that mean? It's basically you're going to basically lose the contact inhibition activity, or the contact inhibition ability, I should say. And when you lose the contact inhibition ability, the cells are going to keep proliferating without control. تمام? So one of the important mechanisms of E-cadherin is how it interacts with the NF2, neurofibromin 2. هذا, it acts downstream of the E-cadherin in a signaling pathway, and in this way it's going to help contact inhibition. But what if a homozygous loss occurs? Then neurofibromatosis type 2 can develop. Okay? تمام. E-cadherin also regulates the contact inhibition through beta-catenin signaling pathway. Okay, let's go down to the picture actually now. Yes, here we go. We kind of have to study the things that are going to come next, يعني. But anyways, here we have the APC gene. This APC gene acts as a protector, okay? Acts as a protector, يعني خلينا نحكي as a tumor suppressor gene. It basically surrounds the beta-catenin and destroys the beta-catenin, not allowing it to enter to the nucleus. Okay, it won't activate specific factors for proliferation. تمام? Now, if the APC gene, gets mutated, okay, the beta-catenin is going to be free to do whatever it wants. It will enter to the cell and it will cause proliferation. So, what does the E-cadherin have to do here? E-cadherin sends a signal to the beta-catenin to allow it to enter and cause proliferation. This is normally, okay? Normally, هيجي signals from what we call a WNT, and this is the WNT receptor, and so on, okay? So we're gonna get to that later when we study the WNT part, okay? Anyways, APC gene mutation, I want to talk about that, in adenomatous polyposis coli. Adenomatous polyposis coli beta-catenin pathway. The APC gene is basically has an anti-proliferative effect in an unusual manner. It's a protein in the cytoplasm. What does it do? It regulates the intracellular levels of beta-catenin. It notices if there is an extra amount of beta-catenin. This means that beta-catenin is going to go to the nucleus and cause proliferation. The APC gene is just going to regulate the amount. If that amount exceeds a specific number, then we gotta destroy them, because this can cause multiple, or a large amount of proliferation that is unneeded. Okay, so really simple. Beta-catenin is a protein with many functions. Beta-catenin binds to the cytoplasmic portion of E-cadherins, which is a cell surface protein. This E-cadherin, it mediates the intercellular interactions. Intercellular يعني interactions between cells, okay? نفس البيتا-catenin, it can translocate itself to the nucleus, يعني بيمسك حاله من الإي-cadherin وبينزل للنوكليوس, okay? How can it do that? Or what is the ability that it has, or what is the ability that makes it do that? Basically, it's an important component of the WNT signaling pathway that regulates the cell proliferation. تمام? WNT, it's a soluble factor that can induce cellular proliferation. Basically, it's a ligand that binds to a receptor, okay, and causes a cellular proliferation. بس بكل بساطة. طيب. What happens when the WNT binds to its receptor? It prevents the APC from degrading the beta-catenin. تمام? So the APC gene here is going to get inactivated. تمام? And it will allow the beta-catenin to enter to the nucleus, or translocate to the nucleus. تمام? So something that is really simple. Basically, شو بيصير? It acts as a transcriptional activator in conjunction with another molecule called TCF. So the beta-catenin binds with the TCF, and it will act as a transcriptional activator. طيب. What about quiescent cells? Quiescent cells are cells that don't want to grow. Basically, they're not exposed to WNT. That's why the cytoplasmic beta-catenin will be degraded by APC, okay? So it's a destruction complex, and APC is a part of it. What if APC is lost? If APC is lost, there is beta-catenin present in the cytoplasm. It will translocate to the nucleus, and it will cause proliferation even to a quiescent cell, apparently, okay? So beta-catenin degradation is prevented in the WNT signaling, in a malignant cell, okay. It's prevented, and the WNT signaling response is inappropriately activated in the absence of WNT itself. يعني the pathway itself is going to be activated without the need of a ligand to bind to the receptor. يعني WNT doesn't even need to bind, okay? This will lead to transcription of growth-promoting genes like cyclin D1 and MYC gene, whatever type of gene, okay? So basically, we can say that the APC gene is a tumor suppressor gene, okay? Because the presence of APC is enough to degrade the beta-catenin, and if it does so, it will prevent the cellular proliferation, the excessive cellular proliferation, okay? So APC is really important. Individuals born with one mutant allele develop hundreds to thousands of adenomatous polyps in the colon during their teens or 20s, which show loss of the other APC allele. Okay, so basically, individuals that are born with one mutant allele, eventually they will lose their other allele, okay? Almost invariably, one or more polyp is going to undergo malignant transformation upon the accumulation of other mutations in the cells within the polyp. Okay, so نفس الperson عم بيحكي لك he will develop hundreds to thousands of adenomatous polyps, and invariably one or more of them is going to undergo malignant transformation. It's going to become a malignant tumor. Okay. And this is not a good thing, obviously. APC mutations are seen in 70 to 80% of sporadic colon cancers. Sporadic, as we said, it occurs in non-familial way, non-inheritable way. Okay, so 70 to 80% of these cases, they have APC mutations. Colonic cancers that have normal APC genes show activating mutations of beta-catenin that render them refractory to degrading of the degrading action of APC. شو اللي عم تحكي انت؟ So we said 70 to 80% of the colon cancers, sporadic, they have APC mutations. So what about the 30 to 20 or 20 to 30% that basically don't have APC mutations? شو مالهم هدول؟ They have normal APC genes, ممتاز. لكن the mutation happens, the mutation happens to the beta-catenin. It will activate the beta-catenin. It will start not caring about the APC. Okay? بتفل من ال APC. The destructive complex. تسحب حالها, told itself, and it goes and translocates to the nucleus. طيب. What about the evasion of apoptosis? Let's know what apoptosis is أول. There are two distinct programs that activate apoptosis. عندك extrinsic pathway and intrinsic pathway. Extrinsic من برا, from the outside, a signal arrived. Intrinsic pathway, something from the inside of the cell. Something is wrong internally. ايش اللي wrong internally? DNA. بس هذه السلافة كلها. طيب. Extrinsic pathway, it's called the death receptor pathway. We're gonna talk about the CD95 اللي هو نفسه الفاس. CD95 is FAS. Okay, they're the same thing. Okay, this is a receptor. It will basically receive a specific ligand called FAS-L. ايش اسم الليجند؟ FAS-L. بدل FAS-L تقدر تحكي CD95L. Okay? And when it binds to the receptor, it causes the trimerization of the receptor. تمام? And basically, the death domains are going to be activated. A domain called death domain alpha. Okay? Anyways, let's start talking about that pathway. The extrinsic pathway is initiated when the CD95, the receptor, is bound to its ligand. Mean the ligand CD95L, FAS-L, causes trimerization of the receptor, as we just said, and thus its cytoplasmic death domains, alpha. Then it will cause it will attract the intracellular adapter protein FADD, FAD, which will recruit pro-caspase 8. شو يعني pro-caspase 8? يعني it's still not ready. Pro-caspase 8, still not ready. To form the death-inducing signaling complex. Okay. Pro-caspase 8 is then activated. كيف activated? By cleavage. I cleave it to smaller subunits, I will generate caspase 8. Caspase 8 should be amended. It will activate downstream caspases such as caspase 3. هذا بنسميه executioner caspase. Okay? So caspase 3 هو executioner caspase. It will cleave DNA and other substrates to cause cell death. حلو. Let's look at the picture and understand it in an easier way. هي الفاس receptors. هي الفاس-L. الفاس-L هو ligand. It basically binds itself to the receptor, caused the trimerization. برضو caused the trimerization of the death domain complex alpha أو death domain alpha. تمام؟ هون شو صار؟ It attracted the FADD. هي ال FADD. FADD caused the secretion of procaspase 8. Procaspase 8 was cleaved into smaller subunits اسمهم caspase 8. Caspase 8 activated caspase 3. هذا ال executioner. It will kill the DNA and other proliferative substrates. تمام؟ It's really easy, nothing difficult one. What about the intrinsic pathway? It's triggered by a variety of stimuli. شو هم? Withdrawal of survival factors. Intrinsic factor هو إنه something in the inside is not correct. Okay? One of them can be withdrawal of survival factor. Survival factor can, when basically they stop arriving to the cell or they stop activating the cell will not find a need to survive. بس بكل بساطة. Stress and injuries from whatever. Stress or injuries may be accumulation of reactive oxygen species, DNA mutations, excessive DNA mutations, maybe some organelles are facing some problems, whatever, sort of injury. So, activation of this pathway will increase the permeability of the mitochondrial outer membrane. حلوين. The mitochondrial mean outer membrane. So it will cause the release of specific molecules منهم cytochrome C. These will initiate apoptosis. هلا نحكي كيف. طبعا we studied all that in cytology. The integrity of the mitochondrial outer membrane is regulated by pro-apoptotic and anti-apoptotic members of the BCL2 family of proteins. شو اللي قاعد بتخبصه؟ بدنا نفهم في عندنا شيء اسمه pro-apoptotic factors and anti-apoptotic factors. Pro-apoptotic means that they will eventually cause apoptosis. Okay, they will cause it. Anti-apoptotic, they eventually will not cause apoptosis. Okay, they will stop. They prevent apoptosis from occurring. Okay. طيب. So the integrity of the mitochondrial outer membrane is regulated by both of them, pro or anti. تمام؟ الانتي-apoptotic هم ال BCL2 family of proteins والpro-apoptotic بنسميهم ال backs and back. Okay? Backs and back. They are required for apoptosis and directly promote mitochondrial permeabilization. تمام؟ طبعا we must understand that in a normal case the mitochondria's outer membrane is not permeable. Okay? طبعا what I mean by permeable here مش permeable أخذناها زمان إنه permeable to specific protein. لا, I'm talking about permeable to everything. Okay, here it's not permeable to everything. Okay, أكيد it has some sort of permeabilization for specific things. Okay, لكن هون we're talking about a normal case for everything. هلا mitochondria it is not permeable for everything. تمام. المهم هلا أكيد there is something that's preventing the activity of the pro-apoptotic factors. ليش؟ the pro-apoptotic factors will increase the permeability of the mitochondria. تمام؟ or mitochondrial outer membrane. طب in the normal case there's something that is basically inhibiting this effect. هم anti-apoptotic. Okay? So the anti-apoptotic factors, they're basically inhibiting the pro-apoptotic factors from increasing the permeability of the outer membrane. تمام؟ مين هم anti-apoptotic هم BCL2 or the BCL-XL. There is a third set of proteins that are called the BH3 only proteins. They're called BAD, BID, and PUMA. Regulate balance between the pro and the anti-apoptotic members. How does it regulate this balance? By its number. تمام? By its number. يعني when the amount of BH3 proteins expressed is high, this means that I'm going to activate the pro-apoptotic members and I'm going to deactivate or inactivate the BCL2 anti-apoptotic members. تمام? So it's just something that depends on the number of proteins of the BH3 family. That's how it regulates it. That's how it balances them. Okay? So when the sum total of all the BH3 proteins expressed overwhelms the anti-apoptotic BCL2 and BCL-XL barrier, the backs and back are going to be activated and they will cause the formation of pores in the mitochondrial membrane. From these pores, the cytochrome C will leak into the cytosol. In the cytosol, the cytochrome C binds to APAF1. Okay, the cytochrome C binds to the APAF1. And these will cause the activation of caspase 9. Caspase 9 kind of has the same ability as caspase 8. It will activate an executioner caspase caspase 3. Because of the pro-apoptotic effect of BH3 proteins, efforts are underway to develop of BH3 mimetic drugs or mimetic drugs. Okay? Scientists are thinking of methods and ways to develop drugs. These drugs are going to act as the BH3. Why will it act as BH3? Because it will cause the overstimulation or overexpression of BH3 on the mitochondrial membrane. This will cause the overwhelming of the anti-apoptotic factors, promoting apoptosis. So why would I ever want to promote apoptosis in a person? Man, we're studying cancers. I want to promote apoptosis in the cancer cells. Okay? Anyways, the malignant cells can escape apoptosis. This is the bad thing, and that's why we want to study and create BH3 mimetic drugs. Anyways, how can malignant cells escape apoptosis? So evasion. Reduced levels of CD95. The same receptor, it has reduced levels of that, اللي هو الFAS receptor. Okay? So basically, the tumor cell, it will render the tumor cells less susceptible to apoptosis by FAS ligand, اللي هو الFAS-L. Okay? So what's going to happen is now the tumor cells, because they do not have as much Fas receptors, the Fas ligand is not going to do anything, okay? So it's going to make the tumor less susceptible to apoptosis. Some tumors have high levels of FLIP. This protein can bind death-inducing signaling complex and prevent the activation of caspase 8, okay? The death-inducing signaling complex المفروض إنه it's something that's going to induce death. لكن the tumors, they have high levels of what's called a flip. شو يعني flip؟ تقلب. Basically, the flip is going to flip the situation from death-inducing that activates caspase 8 and 2H to something that will prevent the activation of caspase 8. Okay, so flip binds to the death-inducing signaling complex, preventing the activation of caspase 8. Really simple. Reduced egress of cytochrome C from mitochondrion as a result of upregulation of BCL2. Here's the problem. The mitochondria itself, for patients with cancer or for cancer cells themselves, cancer cells themselves, the mitochondria here, they have more BCL2 proteins than any normal cell. So basically you would need a very larger amount of BH3 family to do this regulation now. That's why the cancerous cells are going to tend to survive longer, okay? Because the intrinsic pathway مش شغالة أبدا. ليش مش شغالة? Why is it not working, the intrinsic pathway? Because the BH3 proteins are not doing anything about any problems, okay? The BH3 is not comparing, it's not overwhelming the amount of BCL2, تمام? برضو reduced level of pro-apoptotic factors, يعني increased anti-apoptotic factors and reduced pro-apoptotic factors. ليش? Because of the loss of P53. حلو. Loss of APAF1 that binds to cytochrome C activating caspase 9. If APAF1 isn't present, caspase 9 will never be activated. The executioner caspase 3 will never be activated. برضو upregulation of inhibitors of apoptosis, whatever they are, okay? Of all these genes, the role of BCL2 in protecting tumor cells from apoptosis is best established. Okay? And from all the genes, the role of BCL2 in protecting the tumor cells is best established. From all the mistakes and all the things we just talked about here, okay? The thing that has the largest effect or prevents apoptosis or prevents tumor cells from apoptosis that protects tumor cells from apoptosis هو ال BCL2. Okay? 85% of B-cell lymphomas of the follicular type carry a characteristic chromosome 14-18, position 32-21 translocation. Okay? Chromosome 14q32, the site where immunoglobulin heavy chain genes are found. It's also involved in the pathogenesis of Burkitt's lymphoma. طيب, what am I talking about here? B-cell lymphomas of the follicular type حصير فيها translocation from chromosome 14 to chromosome 18, position 32-21. Okay? Really simple. They're just talking about the translocation here. Anyways, juxtaposition of this transcriptionally active locus with BCL2 that is located at chromosome 18, position 21, causes overexpression of the BCL2 protein. Just how we mentioned in I think a couple lectures ago about the fusion genes and translocation. Anyways, basically the BCL2 gene خلينا نحكي حيصير في overexpression of the BCL2 protein. Okay, it's a really simple concept. The BCL2 is going to be overexpressed. Why did it get overexpressed? Because of this translocation. شو translocation? It's a mutation. Okay, so again, let's just make it simpler. Chromosome 14, chromosome 18, a translocation occurs. When the translocation occurs, the BCL2 protein is going to get overexpressed. شو يعني? You're going to find it on the mitochondria more. Okay, more BCL2 يعني more protection from apoptosis. تمام? That's it. This in turn increases the BCL2 buffer protecting lymphocytes from apoptosis and allowing them to survive for longer periods. Steady accumulation of B lymphocytes results in lymphadenopathy and marrow infiltration. BCL2 overexpressing lymphomas arise in large parts from reduced cell death rather than explosive cell proliferation. They tend to be indolent, slow growing compared with many other lymphomas. يعني here these lymphomas or the BCL2 overexpressing lymphomas, basically the reason they arise is due to the reduced cell death. مش من وراء the proliferation, because the proliferation is slow here, okay? They have slow growing. When I'm talking about the lymphomas, they have slow growing. But what is the thing that makes them cancer? It's basically the reduced cell death. They have a prolonged cell life. They have prolonged life, okay? And this is the bad thing here. Limitless replicative potential. Most normal human cells, they have a capacity of 60 to 70 doublings. Basically, they can double 60 to 70 times. يعني. After this, the cells will lose the capacity to divide and they will enter senescence. This phenomenon is due to the progressive shortening of telomeres at the end of chromosomes. طيب, شو هذا التخبيص كله? Here, this is a chromosome. Huh? Chromosomes, طبعا, شو هم عبارة عن DNA. They're basically DNA. DNA is double-stranded. Okay, so this is double-stranded DNA, double-stranded DNA, double-stranded DNA, double-stranded DNA. تمام? These double-stranded DNAs, what do they have? They have single-stranded ends. هيك. تمام? They have single-stranded ends. These ends are called telomeres. Telomeres. Okay, telomeres means end أصلا. Telomeres. Okay. When DNA replication occurs, basically you're putting in some sort of danger on the cell. With each replication or with each DNA replication, part of the telomere is gone. Hello, okay. Part of the telomere is gone. Here I'm talking about a normal cell, by the way, okay? Eventually, with multiple doublings, all the telomere chain is gonna go. شو بضل? You're gonna be left with a normal chromosome. Eventually, any more doublings are gonna cause deletion mutations. ليش? Because you're gonna start losing genetic material. تمام? This is not something wanted. That's why the cells will enter senescence. لما أنا أوصل لهون خلاص telomeres are gone, the cells لحالهم بيدخلوا senescence, or else deletion mutations will occur. Anyways, شو الفكرة هون؟ طب شو الموضوع؟ شو دخل لي أنا هذا الموضوع؟ Tumor cells, they have overactivated telomerase activity. شو اللي telomerase؟ شو اللي قاعد بتحكي فيه أنت؟ We took this in توجيهي يعني if you remember it. طبعا this is the chromosome. Telomerase is basically the enzyme that is going to keep the telomere chain long. This telomerase is actually overexpressed in two types of cells. Stem cells وبرضه cancer cells. بطلت أعرف أكتب. حلو. ما حد يدرس الصوبة عنده وخاصة ما حد يصور محاضرة والصوبة عنده. أنا انخنقت صراحة. Okay. From the carbon monoxide. Okay. Anyways. Stem cells and cancer cells, they have overexpressed telomerase activity. تمام? And due to the overactivity, the telomere chain is going to be longer. This means that no matter how many proliferations occur, more than 60, more than 70, more than 100, the telomerase is going to grow the chain again. طب شيل كمان مرة, the telomerase is going to grow the chain again. تمام? So that's why the cancer And that is why the cancer cells, they tend to live longer. لأنه telomerase is always active, elongating the telomere chain. تمام? هلا, let's take a look at what happens when telomere, راح نختصر الحكي فوق كله, نعيده يعني. What happens when the telomerase is inactivated? In normal cells يعني. خلصت السل, it's differentiated. It's going to undergo 50 to 70 divisions. Then the telomeres are going to be shortened. The shortening of the telomeres can be detected by a normal P53, by tumor suppressor genes in general. Okay, it can be detected by the P53 method. Okay, at specific checkpoints. If it's detected by our normal system, شو بيصير? I will basically cause the cell to enter senescence. Okay. I don't want the cell to die. I want it to stay alive, but I do not want it to proliferate anymore. So I put it in senescence. طيب. What if P53 is mutated? What if the defense mechanism is mutated? Does that mean you're done? Does that mean cancer arises instantly? لا. There are lots of protective measures inside of our bodies. Okay? The body is going to undergo a specific way called the non-homologous end joining pathway. the NHEJ pathway. This pathway, it will join chromosomes together. It will join ends non-homologously. شو يعني non-homologous? يعني not the same chromosomes, okay? Non-homologous end joining. This end to this end مثلا. And this end to this end. عادي ممكن يكون العكس مش مهم, okay? Anyways, this end to this end or this end, it doesn't matter, okay? Just to make it simple. This will create dicentric chromosomes. شو يعني dicentric? يعني it has two centromeres. This is the first one, this is the second one. It will undergo normal mitosis, metaphase, anaphase. And after anaphase, يعني when anaphase, we're going to split them apart. هيطلع معاك نفس الشيء new breaks. Now new breaks have formed. The NHEJ pathway is going to occur again. It's going to happen again. Why is it going to happen again? This pathway happens so it basically causes something called a mitotic catastrophe. Okay, a mitotic catastrophe. When this pathway keeps on happening, it will not make sense anymore. Okay, something is joining, something is being taken apart, and so on. The dicentric chromosomes, التخبيص هذا كله. So eventually, mitotic catastrophe will occur and the cell will die. Okay? Here comes, خلينا نحكي, the bad part of cancer. If the cell somehow finds the ability to reactivate telomerase, it will start growing the telomere chain again, and the cells will come back to life as cancer cells. Okay? Really simple thing. جد كتير موضوع بسيط, ممكن لما نقرأه معك واضح, لكن مع الشرح إن شاء الله يكون واضح. Okay? Anyways, so the markedly shortened telomeres are recognized. حكيناها, it's a protective measure عشان ندخل senescence. لكن when mutations occur, non-homologous end joining pathways activated in a last effort. تمام. Here the same thing, I repeated it by accident, my bad. So short telomeres are recognized in cells in which the check. Okay, this inappropriately activated repair system results in dicentric chromosomes. They're pulled apart at anaphase, resulting in new double-stranded DNA breaks. Breaks, and the DNA itself is broken. The resulting genomic instability, it will basically form a bridge fusion breakage cycle, eventually produces a catastrophe, a mitotic catastrophe, characterized by massive cell death. Okay? So, basically, tumors that grow indefinitely as they often do, loss of growth restraints is not enough. Tumor cells must develop ways to avoid both cellular senescence and mitotic catastrophe. Here, this is the bad thing. They will find a way to activate the telomerase again, okay? If during a crisis a cell manages to reactivate telomerase, the bridge fusion breaker cycle ceases and the cell is able to avoid death, okay? During this period of genomic instability that precedes telomerase activation, numerous mutations could accumulate. Passage through a period of genomic instability probably explains the complex karyotypes frequently seen in human carcinomas. That's why you'd see different sorts of nuclei and different whatever in cancers, due to the genomic instability that happened. Okay, telomerase active in normal stem cells is normally absent in somatic cells. Telomere maintenance is seen in virtually all types of cancers, and 85 to 95% of cancers due to upregulation of the enzyme telomerase. A few tumors use other mechanisms termed alternative lengthening of telomeres, which probably depend on DNA recombination. In the progression from basically instead of using telomerase, it basically uses DNA recombination حتى تطول التلومير. In the progression from colonic adenoma to adenocarcinoma, from benign to malignant, early lesions had a high degree of genomic instability with low telomerase expression. Okay, so basically في genomic instability. Malignant lesions had complex karyotypes with high levels of telomerase activity consistent with a model of telomere-driven tumorigenesis in human cancer. So, if there was a colonic adenoma progressed to a colonic adenocarcinoma, early lesions, they had a degree of genomic instability, لكن telomerase activity is low. Malignant lesions had complex karyotypes with high levels, what they noticed in patients, not that big of a deal. We explained all three diagrams, and good luck, doctors. Now let's solve some questions. How does the transforming growth factor beta signaling get disrupted in many cancers? هنشوف. Loss of tumor suppressor genes that regulate cell cycle, overexpression of growth factors that stimulate proliferation, mutations in oncogenes that promote cell growth, mutations affecting TGF-beta receptors or the SMAD molecules. The answer here is D, because the signaling تاعها depends on the receptor or the SMAD. What is the primary role of the transforming growth factor beta in normal cells? It is to inhibit cellular proliferation. طبعا obviously يعني I guess this question is kind of not that good due to the answer of immune response. It has to do with immune response يعني. Anyways, just focus on the part that's needed from us to inhibit cellular proliferation. What initiates the extrinsic pathway of apoptosis? The binding of the binding of the CD9 the receptor to its ligand. Okay, the extrinsic factor CD95 CD95L. Okay. How do pro-apoptotic proteins like BAX and BAC contribute to apoptosis? هدول بيعملوا mitochondrial permeabilization. تمام. What is the effect of overexpressing BCL2 in lymphomas? Overexpressing BCL2, it will basically cause increased survival of lymphocytes. What is significance of MYC in relation to TGF-beta signaling? MYC is related to growth. تمام. So basically, MYC is overexpressed when TGF-beta is impaired. So, how does the presence of cytochrome C trigger apoptosis? حكينا binds to APAF1, activating caspase 9. What mechanisms allow tumor cells to achieve limitless replicative potential اللي هي reactivation of telomerase? How does the activation of caspase 9 differ from that of caspase 8? كل واحدة لها pathway معينة. The caspase 8 is activated by cleavage, or caspase 9. Yeah, I guess you can see, you can say the answer is C, no? Like caspase 8 is activated by cleavage. Caspase 9 activated by C binding to APAF1, sorry. What is the connection between telomerase activity and cancer progression? Increased telomerase activity promotes cancer progression. How does neurofibromin or NF2 contribute to contact inhibition? حكينا it has to do with اللي هي الE cadherin, okay? So it's a signaling pathway, acts in a signaling pathway to maintain contact inhibition. Does it enhance tumor growth? لا. Does it promote cellular proliferation? لا. Does it facilitate cell migration? شو دخل? So this. تمام. Good luck, doctors. The exams are really soon now, 15 days. I really wish you the best. شدوا حيلكم بالتوفيق.